Hi students. I've gotten a couple of comments that people have struggled with the last few worksheets. And so what I decided to do is to go over some of them and just kind of work through and talk through the logic of how I would work through those. And hopefully that will help you if you're struggling to understand and apply this material on all by yourself. So first I'd like to go over the complex combinations in the lac operon worksheet. So in 11 through 15, these are pretty challenging. The first thing that I would look at in these problems is where you have functioning genes. And so this first copy of the lac operon, you have to transcribe um, in order to get the Y protein produced. The Z is mutant here, but the Y is wild type, and over here the Y is mutant. And so you must transcribe here to get Y produced, and you must transcribe here to get Z produced. So then the next question is, how is the regulation affected? So in this first operon, we have an constitutive operator, and a constitutive operator means that that is always transcribed. It's always on. And so the Y gene should always be turned on, even in the presence or in the absence of lactose. For the Z gene, here you have a mutant promoter, and if you don't have a functioning promoter, you can't transcribe, and so those ones would be minus. This highlights the fact that the operator and the promoter are both elements that act in cis. That means they only affect the genes that are right next to it on the chromosome. For number 12, here we have Z and Y are both functional in the first one, but only Z is functional here. So the first copy of the operon has a mutant promoter, and a mutant promoter, there's nothing you can do to ever transcribe this copy. And so this is the only good copy of the Y gene, and so Y will never be able to be transcribed. You can't transcribe this at all, but you can transcribe this one over here, and so Z could be produced from the second copy of the operon. Here you have a good promoter, you have a good operator, but you have a mutant repressor. However, the repressor will act in trans, and so the repressor can come over, be encoded over here on this copy of the operon, but then that protein can bind to any functioning operator, and this copy of the operon would actually be transcribed as usual, so the same as wild type, in which case the repressor would bind to the operator when lactose was absent, and so it would block transcription, and when lactose was present, the repressor would fall off, and then you would be able to transcribe. For number 13, let's look at this. So we have the functional Y gene. Here we have a functional Z gene here. So here we have a good promoter, a good operator, but we have a super repressor. And a super repressor will actually that's a version of the repressor that can still bind to the DNA, but it cannot respond to lactose by changing its conformation and falling off of the DNA. So it no longer binds to lactose. So that super repressor would bind to that operator and would just turn that copy of the lac operon off. And so this one would be Y, and so here would be negative for Y. Over here, we have the good Z gene. We have a good promoter, good operator, and we have a mutant repressor. Remember that the repressor acts in cis, and so that super repressor can actually come and bind to that operator, and in that case it would permanently kind of turn that one off as well, and so you wouldn't get any transcription from that one. For number 14, you have a good Y gene on that first one, you have a good Z gene on the second one. So here you have another super repressor, and in this first copy of the operon, that super repressor would bind to that operator and inhibit transcription from that copy of the operon. And so for the Y gene, which is only encoded here on that first copy, it would be turned off all the time. On the other copy, you have the functioning Z gene, but you also have a constitutive operator. And so in this case, the repressor can act in cis, and you would normally think that a super repressor could come over and bind over here and repress that. However, it actually doesn't because you have that OC mutation. And so it doesn't matter what kind of repressor you've got, it can't bind to that OC mutation. And so you have a good promoter and you would constitutively activate the Z gene. Last one, number 15, you have Z and Y are both functional here, and these are both mutant. So you need to transcribe this one in order to get both of those genes produced. Here you have a mutant promoter, and you have also a mutant operator or constitutive operator. So these would normally give opposite phenotypes, these two mutations. The one that wins out here is the promoter mutation. So the promoter, if you can't get RNA polymerase bound, you cannot transcribe that gene, and you would never be able to transcribe that copy of the operon. And so that would be negative all the way across. Next, I'd like to go over the GAL system worksheet. And so I'll start off by just doing a little review of what this operon looks like. So the GAL genes, the GAL 1, 2, 7, and 10 genes, all are separate genes on different regions of the chromosome, and they each have their own regulation system. So each one of these would have a core promoter, and each one of these also has two binding sites. So the very first binding site I'll talk about is the UAS sequence, which is the binding site for the protein GAL4. And GAL4 is the transcriptional activator for this particular gene. And GAL4 gets inhibited by another protein that binds and kind of inhibits its function, and that particular gene is called, or protein is called GAL80. And then there's a third part to this, and this one would be GAL3. And when you have GAL3, GAL3 is the thing that binds to the sugar galactose. So if that little black dot's galactose, then when GAL3 binds the sugar, it actually pulls GAL80 off of GAL4, and it increases the affinity of GAL80 for GAL3. And so GAL80 will just move between this GAL3 protein and the GAL4 protein, in responding to whether galactose is present. The second major system here is the system that responds to glucose in the cell. And so this is a second binding site for a transcription factor. This would be the MIG1 binding site. And to that sequence, MIG1 would bind, and MIG1 recruits another protein called TUP1, and TUP1 is a co-repressor that helps to turn off those genes. Now, we're going to consider different cases. So in the wild type, if you are lacking glucose and also lacking galactose, what would happen? So if you have low glucose, that means that MIG1 will not be transcribed, and so that protein would not be present. And if you are lacking, or if you're lacking galactose in that cell, then GAL80 would be bound onto GAL4, because GAL80 only gets pulled off of GAL4 to bind with GAL3 when galactose is present. Okay, so in that first case, GAL80 is inhibiting GAL4, and these proteins would not be recruited to that part of the promoter. And so the overall effect of that would be that these GAL structural genes would not be transcribed. So we would say that one's negative. If then glucose is still absent, but galactose is added, then what happens then is galactose binds to GAL3 and GAL80 gets pulled off, and GAL4 is now activated and able to transactivate uh, transcription of those genes. Glucose is not present, and so the MIG1 and TUP1 are not bound to that region, and so these genes would be turned on. Finally, the last case, when you add glucose and you also have galactose. So first part, when you have galactose, the GAL80 is pulled off and it's bound over here to GAL3. And so you would think that GAL4 would be active. This is the transcriptional activator and would be able to activate transcription of those genes. But it actually doesn't, because glucose is also present in the cell, and when glucose is high, MIG1 gets turned on. And so this protein MIG1 will bind to the DNA of the enhancer region right next to these GAL structural genes, and it will recruit this factor called TUP1. Now, TUP1 is a histone deacetylase. It would be considered a transcriptional repressor. And what will happen is that if TUP1 is bound to this region of the genome, it actually overrides what happens with GAL4, and it would turn off transcription, sort of dominantly turn off transcription of those structural genes. And so the result of that is that you would be turned off at that point. So let's talk about these next ones. So GAL80, what if you had a mutation in GAL80? So if you were lacking GAL80, then there's no way for GAL4 to be inhibited by this particular, by this normal mechanism of regulation by whether galactose is present or not. And so when you are missing galactose in the cell, you would actually not be able to turn that off. GAL4 would be activating those genes. Also, for the next one, that would be normal, so GAL4 would still be active. And however, in that third column, you add glucose, and glucose, this particular regulation mechanism is still in play, is still working. And so if you add glucose, MIG1 gets turned on, it recruits TUP1, and this one will dominantly still inhibit the GAL4 transcriptional activator, and it would turn off transcription of those genes. If you're lacking GAL4, so number 3, then there is actually no way that you can turn on transcription of these genes. So GAL4 is absolutely required to transcribe this. So you wouldn't be able to turn it on in any of those cases. What if you had a GAL3 mutant? You actually don't have the thing that would be able to respond to galactose in this case. The result of that would be that GAL80 would always be binding to the GAL4 protein, and it would always be inhibiting it. And so here you would also be negative all the way across. What if you had a mutation in the UAS enhancer sequences for the GAL1 gene? So what I'd like to do in this case is answer this separately for the different genes. So for GAL1, we don't have a functional binding site for the transcriptional activator, and so you can never recruit that transcriptional activator to that region of the genome, and you can never activate the GAL1 gene. And so this would be negative all the way across. But the UAS enhancer sequence is a cis-acting factor, so it is only going to affect the gene that it is right next to. And so in this case, that mutation was in the UAS enhancer sequence next to the GAL1 gene. The GAL2, 7, and 10 genes should be just fine, and they will in fact function just like wild type. There's no problem with how those ones are working. And so this is just like we observed in wild type. If we had a mutation in MIG1, then what would happen? So this system with galactose would be still functional because MIG1 is this glucose responsive element, and so this part would be just like wild type when we responded to galactose. However, if MIG1 cannot be However, if MIG1 does not increase in response to glucose added to the environment, then what would happen is you can never recruit that transcriptional co-repressor TUP1, and you wouldn't be able to kind of override what's going on at the other in the other regulation system. And so GAL4 in this third column would still actually be able to it would be active since galactose is present, so GAL80 was pulled off of GAL4, and this would actually be incorrectly active. We don't have this thing that kind of overrides it and makes it negative. MIG1 constitutive, so MIG1 is not responsive to glucose, it is always high. So if that happened, then no matter what was happening over here, we know that MIG1 and TUP1 are actually dominant over the effect of what's happening with GAL4. And so if MIG1 is always there, it's always going to recruit TUP1 to that region of the genome, and you will always turn off those genes nearby. And so that would be negative all the way across. Then finally, if you had a mutation in TUP1, you would just be lacking that co-repressor that binds to MIG1. And so this would really be the same effect that we observed with a mutation in MIG1, which is the galactose system is functioning normally, but you cannot actually turn off this. The presence of glucose does not result in the dominant kind of turning off of these genes, and so it would be, it would be positive there as well. It wouldn't be able to inhibit what's happening with GAL4 because we don't have recruitment of this co-repressor. The last worksheet I'd like to go over is this transcriptional regulation review of the LAC and the GAL systems. So in this worksheet, what I'm really trying to do is help you take the things that were identified in the LAC and the GAL systems and to kind of categorize them according to how we name things in this field of transcriptional regulation. So it's really important to pay attention to this first part, which is kind of the definitions of what these different things are. So a transcription factor, first of all, must be a protein. It must be a protein that binds directly to a DNA sequence near a gene, and it has to function to either help to activate or to repress transcription of that gene right nearby. An effector is actually not a protein. This would be a small molecule from the environment that the cell somehow senses and responds to. And so these are often used to regulate the activity or the function of proteins. Then we have transcriptional co-activators and transcriptional co-repressors. So these would both be proteins as well, and these would be proteins that affect transcription. They get recruited to a region of the genome, but they do not bind directly to the DNA. So this would be something that generally binds to another transcription factor. So the transcription factor comes in first, binds to the DNA, and then it recruits a transcriptional co-activator or co-repressor. Transcriptional co-activators and co-repressors are generally both present in the cell all the time. They need to be recruited to a particular region of the genome in order to affect transcription of that region of the genome. They don't just broadly affect transcription of the whole genome at the same time. And then finally, cis-regulatory elements. This includes enhancers and promoter proximal elements. Cis-regulatory elements are the binding sites for transcription factors. And so once we identify transcription factors in a system, the very easy way to identify the cis-regulatory elements would be to think about what are the sequences that those transcription factors bind to. So first let's go into the lac operon. So for the lac operon, we discussed two transcription factors. One of them is a transcriptional activator and one is a transcriptional repressor. So the repressor is the one we started out with. This would be called lac-I, or often we just call it the lac repressor. The other one is the protein called CAP. And so CAP is the protein that binds to CAMP and responds to the levels of glucose in the cell. So let's just briefly mention then what the mechanism of action of each of these factors is. So for lac-I, or the lac repressor, it sits on the operator and it physically blocks transcription from happening. So it sits right where you would start transcription, and it prevents RNA polymerase from getting in there and actually starting to make a messenger RNA. For CAP, its mechanism of action is actually to bind to the DNA right next to the promoter and to help recruit RNA polymerase more rapidly. There are two cis-regulatory elements in the lac operon. These would be the binding sites for these two transcription factors. And so the binding site for the lac repressor would be the operator, and the binding site for CAP is actually just a sequence right next to the promoter. It doesn't really have a specific name. We're just going to call it the CAP binding site. And then there are two different effector molecules that function in lac operon regulation. The first one would be lactose itself, the sugar lactose. So lactose binds to that lac repressor and makes it change its conformation, and it changes its affinity, changes the affinity of the lac repressor for the DNA. But then the other one is actually kind of a combination. I'll write them both, glucose and CAMP. And so specifically CAMP is oppositely regulated as glucose. So if glucose is high, CAMP is low. And these would both be considered effectors in the lac operon. For the GAL system, there are two transcription factors that regulate in this system. So these would be proteins that bind directly to DNA to help regulate transcription. So the first one would be the GAL4 protein. This would be a transcriptional activator. And the second one would be MIG1. So what is the mechanism by which it accomplishes its action, or it accomplishes its job? So GAL4 does a huge variety of different things in order to activate transcription, and it's kind of a sequential order of events. So one of the first things that happens is it recruits a histone acetyltransferase, or a HAT, in order to help in order to help acetylate the histones in that region of the genome. And then it recruits a chromatin remodeling complex called SAGA to move the nucleosomes out of the way to kind of expose the core promoter. And then it also helps to recruit the basal transcriptional apparatus, first by interacting with a protein called mediator, which helps to recruit RNA polymerase to the promoter. And also it interacts with a general transcription factor called TF2D, which then helps recruit the basal transcriptional apparatus. MIG1 accomplishes its job by recruiting a co-repressor, and that co-repressor would be TUP1. And TUP1 is a histone deacetylase, and so that histone deacetylase removes the acetyl groups from the nucleosomes in that region of the genome, thereby turning off that gene. What are the cis-regulatory elements which function in the GAL system? So the cis-regulatory elements are the binding sites for the two transcription factors that we identified in number 4. And the first one is called a UAS enhancer. That would be the binding site for GAL4. And then the second one would be the MIG1 binding site. And these would both be considered enhancers sequences. Next, what are the two effector molecules that function in the GAL system? One of those would be galactose, the sugar galactose, and the other one would be the sugar glucose. Then finally, there are many co-activators and co-repressors that function in the regulation of the GAL structural genes. So what I'd like you to think of is what are the things that bind to those transcription factors? And we've mentioned quite a few of these already. So first of all, let me start with MIG1 since it's more simple. So the thing that binds to MIG1 is TOP1, and it is a co-repressor, and it does that by histone deacetylase activity. And then the things that bind to GAL4, there are a lot of those. So the SAGA chromatin remodeling complex, there's a histone acetyltransferase, there is mediator, and another thing that you could probably call a co-repressor would be GAL80. So GAL80 is that protein that binds to GAL4 and inhibits its function. And we could also probably call the TF2D a co-activator, although it's really part of the basal transcriptional apparatus.